welcome to Season 2 Purdue University College of Sciences Superheroes of Science podcast. I'm Stephen.
And I'm Sarah. We will be discussing anything and everything related to science. If you have a science question, tweet it to us at Purdue SOS, and we will try and find someone to answer it for you. Today on Superheroes of Science, we have Mike Reppert, Associate Professor in the Department of Chemistry here at Purdue University. So welcome, Mike.
Oh, thank you. Good to be here.
Thanks for agreeing to be interrogated by us today.
Oh, absolutely.
And so let's start with, what is it your group researches?
Yeah, well, my group is generally interested in biophysics and spectroscopy. So especially the most thing we focus on most of the time is photosynthesis, so kind of understanding how green plants and algae and cyanobacteria do what they do, produce oxygen and fuel for the rest of the planet.
Mm-hmm. So, there's like three words that threw up flags that I want to ask
Sure.
about now. Spectroscopy, first of all.
Yeah.
What, can you help us understand
it's sort
that?
of a tricky one. So loosely speaking, spectroscopy is just studying how light interacts with matter. So in photosynthesis in particular, we're interested in how sunlight comes in and interacts with these chlorophyll pigments in photosynthetic proteins to produce all of the stuff that we think of from photosynthesis, growing plants and fuel for us to eat, for other animals to eat, and biofuels even now, things like this. So it's sort of a big picture scheme from sunlight into all the stuff around us that we're used to on a day-to-day basis.
Well, excellent.
So it's not as simple as sun hits it and it just turns into
that's sort of the basic idea, but it's a little bit more complicated than that in the details, as it turns out.
And what's the first thing you said? Biophysics?
So biophysics.
Biophysics, you said.
Yeah.
And so
biophysics is again kind of I do a lot of intersecting in my research. So light and matter, and then biophysics is sort of the intersection between biological problems, in our case especially living cells, the growth of cells, and how they eat and grow and produce energy, in our case from sunlight. And then also physics. So we actually study the quantum dynamics that goes on inside of these photosynthetic organisms. It's actually, again, quite complicated, actually describing that process of light collection and transformation into chemical energy. So there's some quantum dynamics that comes in, quantum physics that comes in, some biological processes that we study. So kind of a mix of a lot of things.
what wavelengths are you
sure.
running? What spectrum are you actually looking at for your research?
So everything we do is in the visible spectrum. So that's between
cool.
about 400 nanometers and about 700 nanometers.
So I missed my guess. I was going to guess infrared in there.
Well, we do actually some infrared as well. That's sort of to characterize the structure of the proteins that we're studying. So the infrared spectroscopy, we use to study local vibrations in the proteins, so kind of watch how the protein jiggles.
Okay.
Nice.
And that tells us something about the structure. So whether it's folded into an alpha helix, kind of a spiral, or a beta sheet, kind of looping back and forth like a line at Disney World, or some other structure that we don't understand yet.
I like those descriptions. It's cool.
Yeah.
So what are we doing with the visible?
Yeah, so the visible is sort of our tool to understand what the chlorophyll molecules are doing.
Okay.
So we use the visible because that's the region of sunlight that, the wavelength of sunlight that the plants use, or cyanobacteria use, to produce chemical energy. So they collect visible radiation. The two big peaks from chlorophyll are up in the sort of the two ends of the spectrum, down at 600 or 700 nanometers and up at 400 nanometers roughly. And there's kind of a little bit less absorption in the middle. So these kind of red and blue ends of the spectrum get absorbed, and we mostly study the red end of the spectrum, so around 700 nanometers, 600 nanometers. And we use that, so we essentially study what happens when we change the photosynthetic proteins, when we make a mutation in the protein, change the arrangement of the chlorophyll molecules, and then we use the light to see what effect that had on the function of the protein.
Okay.
So the idea is to sort of poke the protein and see how it jumps in the light harvesting sense.
And that kind of gets into the question I had. What types of samples then are you preparing, or how do you get the samples to test?
Yeah, so we have two different model systems that we work with. One is E. coli, so this is sort of the workhorse bacteria that most everybody uses to produce proteins in research labs. E. coli is not photosynthetic. So we have to specifically engineer particular proteins into the E. coli, so introduce foreign DNA into the bacteria cells, and that triggers it to produce the proteins that we're interested in.
Okay.
So you can make photosynthetic proteins. You can't actually make chlorophyll in E. coli, or, well, you can actually, but it's hard.
Okay.
we don't make the chlorophyll directly in the E. coli. We produce the proteins and then mix them with the chlorophyll later to produce the complexes we're interested in. That's sort of the easiest case, because E. coli is just easy to work with. It grows fast and it's easy to manipulate its DNA. The more complicated stuff is with cyanobacteria. So these are, you can sort of think of them like E. coli, but with photosynthetic proteins. So they're capable of doing photosynthesis, but the particular strain we're working with is also capable of just growing from glucose. So you could just feed it glucose, and the nice thing for us about that is that we can do whatever we want to the photosynthetic proteins genetically. So we can introduce mutations that in a cell that depended on photosynthesis would just be lethal. The cells would just die. But because the strain we work with can grow on glucose, we can kind of do whatever we want to the photosynthetic apparatus and see what happens, and the cells stay alive long enough for us to characterize what happened.
Right.
Two questions. One, answer first. What is a protein?
Sure.
Because we keep using the word, saying the word protein.
Yeah, it's a good question.
I'd like us maybe define what that means.
Right.
And then end up with, after that, my next question, I'll throw both at you now. Why?
Sure.
Why are you researching this? What's this telling us?
Yeah, so what is a protein is a really good question. It's actually about, let's see, about 100 years ago, everybody was asking that question. So it was, originally people noticed that there was this nitrogen-rich material that seemed to be really important in the function of cells. So if you sort of boiled cells and harvested all the stuff, there was a lot of nitrogen-rich material that seemed to be important for the function of the cell, and they called that protein. At the beginning, nobody knew exactly what that was. Later, we sort of figured out piece by piece that it's a collection of, you can sort of think of like little beads on a chain. There's about 20 different beads called amino acids, and these are individual molecules that can be linked together, just like a necklace, to form long chains that then fold up into particular structures, and it's those structures that do just about everything you can think of to keep an organism alive. So they're the ones that harvest food, they're the ones that process the energy, they're the ones that make and break chemical bonds, they're sort of the workhorses of life.
Is that the same as the protein? I know I'm throwing I want to let you answer the question.
Question.
It is. Now, but what I'm thinking, okay, it's like my breakfast. I tracked my breakfast this morning. I have carbs, proteins, and fats. I try to stay in a balance there. Is that the same protein?
It's the same protein. Yeah, it's the same thing. Yeah, there's all kinds of different kinds of protein. But yeah, protein is the big word that sort of lumps them all together. Chemically, a protein is a sequence of a bunch of amino acids. So it's sort of much like DNA, which is a sequence of nucleic acids. So there's 4 nucleic acids that make up DNA, and there's 20 different amino acids that make up proteins. Yeah. So, and yeah, when you're reading your cereal box, it's exactly that protein that we're interested in.
All right, so then why?
Yeah,
let's go to the why.
Well, so I have to say first that for me the fundamental reason why is just because I really want to know how things work. So I'm just curious.
That's good, yeah.
So when I was a kid, I loved picking apart things and trying to figure out what they did, what was inside of a motor, and this kind of thing. And I think I'm still kind of doing that. But there's also some useful applications in the long run. So what we hope to be able to do is to begin by figuring out how natural photosynthetic systems work, so how plants and cyanobacteria do this in nature, and then use that information to help design new systems, maybe bioengineered strains of cyanobacteria or algae that can be used to produce biofuels more efficiently. So biofuels are kind of a hot topic recently, and the idea here is that you can just grow a plant or an algae culture or something like this in a reactor or even an open field. Corn ethanol, I think, is a good example, where you grow some kind of plant or algae material and then you process it to make diesel or ethanol or hydrogen gas, something like this that can be used to fuel your car or your house or a factory. The big problem with biofuels right now is that the overall process, if you think about taking the energy from the sun and funneling it down into something you can use, like ethanol or hydrogen gas, the efficiency is very, very low. So naturally for plants, this is about 1%, which even compared to what we can do with solar cells right now is quite bad. we're about natural biofuel production is about an order of magnitude 10 times less efficient than typical solar cells that you can buy on the market right now.
Okay. Yeah.
So the idea is, and the reason for this is actually sort of intuitive. It's that plants and cyanobacteria, natural photosynthetic organisms, aren't optimized for efficiency. They're optimized to be competitive with each other. And so it's actually, if I'm a cyanobacteria, it's actually better for me to waste sunlight if it means that my neighbors don't get it, because then they can't compete with me. So there's an old, I like to give an example of, there's this old joke about two runners who encounter a bear, and the one of them takes off running as fast as he can, and the second runner says, "Why are you running? There's no way you can outrun a bear. Why don't you just give up?" And the first one says, "Well, I don't have to outrun the bear. I just have to outrun you." Right? And this is sort of the same idea. From the perspective of a microorganism doing photosynthesis, the only thing better than having all the sunlight I need is to have all the sunlight I need and to keep it from getting to any of my competitors. So there's this sort of natural selection pressure to have lots and lots of light, even if you're not using it very efficiently.
I see. And then that explains a little bit of why you're saying why the biofuels aren't as efficient.
Right, exactly. So then this in the end translates into low efficiency production for biofuels, because natural organisms actually have specific proteins that are designed just to waste energy.
No way.
Yeah, for real. So after you get above about 20% of full sunlight, energy the specific pathways switch on, and you just start burning energy to just get rid of it. And that's partly also for self-preservation of the organism, because you can if you put in too much light, you can sort of short circuit the system and then damage the organism. So there's these sort of natural safety catches in there that just waste energy.
Yeah.
So we're interested in trying to sort of circumvent those safety catches and make organisms that don't use so much light to begin with. And then the principle can work more efficiently for producing biofuels.
So will we get to the point that in the Back to the Future movie where they used the banana, just dropped it in?
I don't know. That'd be great. But we're not quite there yet. I'm afraid my research probably won't produce that directly. Maybe we can, a grain of sand or something like that toward the pile. Yeah.
I'd ask,
Yeah.
So then, so biophysics then, so what sorts of physics?
Yeah, right. So there's, fundamentally there's all kinds of physics in biology. And for me, there's actually a few different branches of biophysics. So there's people who study sort of system biophysics, which is about sort of using physical ideas to understand how organisms interact with each other and with their environment. And then there's also molecular biophysics, which is more where I'm involved.
Okay.
And that's using physical principles to understand how different molecules interact with each other in the cell, or how proteins fold. So I mentioned before, proteins are these long chains of amino acids. And it's sort of a magical process, as far as I'm concerned, that when you produce a protein with a given sequence, it almost always, well, I shouldn't say almost always in natural systems, folds into this nice, stable, well-defined structure, so that the sequence of amino acids determines the structure of the folded protein.
Okay.
And it's that structure that enables it to do specific tasks in the cell. And this is sort of in my opinion, this is just amazing, that just by determining that sequence of amino acids, it's the same 20 amino acids in all of the millions of different proteins that are out there. It's just by changing the sequence you can get it to fold into long rods or big globs or all kinds of different shapes, almost any shape you can think of. You can do it just by changing that sequence of amino acids. And at the core, that's all determined by the physical interactions between those amino acids. So some amino acids like to interact with other particular amino acids and don't like to interact with others. Some of them like to interact with the water. Some of them like to be shielded from water. So there's all these sort of complicated driving forces that translate that sequence into a structure of the folded protein.
Wow.
So that's one example of where physics comes in, and really hard physics, actually.
It's complicated,
Yeah.
So then are you able to use that knowledge you had mentioned earlier, kind of engineering some of these molecules then to do what you want them to do in the lab. So does that sort of feed in then?
Yeah,
to that?
that's what we're trying to do.
Okay.
I should say I just started last year, so
Okay.
on this project. So we haven't made a whole lot of progress yet. But,
Wow.
but that's what we're working on. Yeah. So the idea is what we'd like to do is take a native protein, native photosynthetic protein, and go in and make specific mutations. That means replacing one of those amino acids that's in the native protein with a different amino acid. And just swapping out amino acids one by one, we, in principle, should be able to change the optical properties, the spectroscopic properties, so which wavelengths of light get absorbed, how those, and the energy gets transferred from site to site within the protein to, in principle, produce better light harvesting, or at least different light harvesting.
Right.
Hopefully better.
But how in the world do you do that? How do you pull one out to put another in?
Yeah, it's biotechnology
this is
amazing.
Totally beyond my understanding.
Right. Well, so I should say I was, when I started this project, I was amazed to realize that you can actually, most of what we can do, you could actually just pay somebody to do. You can literally fill out a form online and submit it to a company somewhere halfway across the world, and they'll actually just do it for you and send you the plasmid that codes for the amino acid sequence that or the DNA sequence that codes for the amino acid sequence that you're interested in. I mean, it's actually not very expensive. The short answer is you can just pay somebody to do it for you. but the longer answer is there's a whole host of tools that have been built up over many years in biotechnology and biochemistry and molecular biology, where there are specific enzymes, often natural enzymes. Again, these are proteins that are able to cleave DNA at particular sites, or insert a little bit of DNA into a place where you're in a site that you're interested in, and so you can go out and synthetically construct the sequence of nucleic acids that you need to start the process, and then you can insert that sequence into the protein where you want to make the change. Essentially, the short version.
Wow.
Yeah, so it's like it's all kind of magical actually, but it works.
That's what it sounds like.
That just made me think of another.
Yeah.
So just thinking of our middle and high school students, it might be. So
Yeah.
you've explained what a protein is and what's so how do enzymes? How are they related to?
Right. Well, so first of all, enzymes are proteins.
Okay.
So like I said earlier, proteins are sort of the workhorse of the cell, right?
Mm-hmm.
So they're the ones that do everything
Okay.
almost. The sort of one exception is nucleic acids are what store information.
Okay.
So DNA stores information, and that information gets translated into a protein,
Right.
and then that protein does some given function. So an enzyme is a particular class of protein that catalyzes chemical reactions. So one thing that protein enzymes, for example, in photosynthesis, are responsible for converting carbon dioxide from the air into glucose that the cell can use for respiration. But another thing enzymes do, there's a whole bunch of different enzymes that are responsible for breaking and making bonds between nucleic acids. So you can, so the cell uses enzymes to replicate the DNA, so to produce more DNA, make a copy of the DNA from what's already there, or to transcribe DNA into RNA, which gets translated into protein. So there's enzymes involved at each of these steps to induce the chemical reactions needed to make that process work.
That's great.
Yeah.
So can you take these basic enzymes and just recreate any kind of protein then, just by putting them in the right sequence?
Well, so in principle, yeah. So you can what you can do, what's relatively easy to do, is if you know the structure of a protein, the sequence of a protein that you want, it's pretty easy to make. To use the enzymes that we have now, well, a combination of synthetic stringing together amino acids. So now you can just do, in the lab you can also just do some of those reactions synthetically. So without enzymes you can string together a bunch of nucleic acids that'll code for a protein. But you use the enzymes then to sort of insert that sequence into the cell's DNA at a place where it'll start actually producing the protein.
Okay.
What's hard is, it's still really an unsolved question, is how do you design a protein to do a particular function? So if you don't already have a protein model that you're starting from, how would you build a protein sort of ab initio from your head? How would you imagine a protein that would do an interesting reaction? We're still a long ways from understanding how you would do that. So in our case too, we're starting with natural proteins, photosynthetic proteins, and sort of tweaking them to make the changes we want. It would be a much harder problem to say, well, we're just going to invent a new protein.
Start from scratch.
Yeah, we're just going to start from scratch. That would be very difficult. Hopefully, eventually, we'll be able to do that too, but not yet.
So this type of research, what are other applications? I mean, you're focusing on like biofuels.
Mm-hmm. Right.
What are other applications
Sure.
that similar people are using this type of process for?
Well, so there's similar research being done. In principle, the kind of work we're doing is applicable to anything that does photosynthesis, or in particular the systems we're working at, anything that generates oxygen in photosynthesis. So that means green plants, that means algae, cyanobacteria. So another application where people are actively working is making crops that yield better, that give higher yields. So there's a couple of examples recently reported where people have been able to make plants grow faster just by messing with these photosynthetic proteins.
So we'll get to the point that I can have a bamboo forest in my backyard.
Well,
'cause I really want that.
We'll see. Well, yeah, you have to think about also whether you want a bamboo forest in your backyard. So that's something you also have to keep in mind with these projects. Is you don't want to mess with the organism too much that it's difficult to control. So
fair enough.
It's also a key thing to think about.
And I guess that kind of brings us into, because a lot of what we're talking about is like genetically modified,
That's right. That's right. Yeah.
And
exactly.
so, and then which is like I don't know if it's I think a trigger word.
Right, sure,
Because
yeah.
a lot of times we don't understand what it means.
Right. Sure.
But so what kind of, I guess I want to say ethics
Sure.
are
Yeah. No,
that's a
involved
great
with
question.
what we're doing? What kind of
Yeah,
ethics are involved with what you're doing that you have to be careful not to cross lines?
Right. Yeah. Yeah. I mean, so I should say, there's a whole host of ethical issues related to bioengineering and recombinant technology. So first I'll say I'm not going anywhere near animals or people. So so just we don't have to worry about that host of issues yet. But yeah, so I think the big issue for us is the question of how do you produce engineered organisms that are safe. It's not so much a question of would they hurt people directly, so like the organisms we work with don't cause disease, and it's sort of hard to imagine how they could cause disease. But what they could do is disrupt the native ecosystem. So there's natural cyanobacteria out there that already perform important jobs. You wouldn't want to create some sort of super synthetic strain that can go out there and just erase all of the natural systems by outcompeting. Like we talked earlier about competition. So you don't want to make something that's so competitive that it's going to go out there and destroy the natural ecosystem. So that's actually one of the things I like about the approach we're taking is that it's actually a way to, the organisms we engineer are actually less competitive overall than natural organisms. I gave the example earlier about bacteria competing for light. So we're actually creating organisms that are less competitive when grown in wild conditions, and will only be competitive when grown sort of in a closed bioreactor or controlled conditions. So the idea there is to try to keep things designed in such a way that if the organism escapes, it just dies. So you don't have to worry about it spreading through the natural system.
Interesting. Yeah.
The example, I grew up on a farm in Kansas, and the example here I like to give is with turkeys. So we raised turkeys growing up, and there were wild turkeys in the fields outside, and there were domestic turkeys here on our farm. And we never worried about the wild turkeys and domestic turkeys mixing, because the wild turkeys were much fitter to survive in the wild than the domestic turkeys. So you didn't have to worry too much about the two breeds mingling. So, yeah.
Well, as a pool of knowledge, who keeps that in check? I mean, could I be the standard mad scientist or something?
Right.
I'm going to generate. Is it possible that person could exist,
Right.
or does the scientific community itself?
Well, this is kind of a debate I think that the community is having right now. Is exactly what. So at a large level, there are some government regulations in place that restrict what you can do. In Purdue, in particular, there's a bioethic or a biosafety committee that reviews. So everything that I do, I have to get passed through a biosafety committee to check this. Is this a reasonable thing to do? Is it going to cause trouble? So in the university setting, there are checks in place to make sure that people are doing things in a reasonable way. Beyond that, it's a little tricky though, because we don't have established. There are a lot of decisions I think left to be made about what constitutes good practice for genetic engineering.
So there's not like an international agreement yet?
I don't think there's any international agreement yet. I think each country has its own guidelines.
I just assumed there was.
Yeah. Right. Yeah, you'd think so, but not yet. At least none very specific guidelines. So you'll see. you'll see headlines every now and then from people in other countries who are doing things that would be prohibited in the U.S. and and that always sparks controversy about is this a good idea or not, and should there be more restrictions in place, which is, I think, a healthy discussion to have.
Absolutely.
Definitely.
Yeah.
Now, if something, let's say another country, a random country did do something, and we know that we look at that organism and we're like, okay, this plant is really going to be so invasive.
Right.
And it's going to disrupt our ecosystems. Does the U.S. itself have laws in place that says, no, you may not import that?
Yeah.
Or is that just like the state of
think-
California only?
No,
Because then you can't get anything across those lines.
Right. Yeah, I think importation across borders is fairly well controlled. Although, in the case of microorganisms, the concern would be that if you made something that can spread without,
Yeah.
okay, the coronavirus is on the
Yeah.
is in the news right now, right?
Sure.
So that's the sort of concern that if you made some microorganism that was really contagious or really hard to contain, it might get imported despite our best efforts. But I should say for the vast majority of genetic engineering processes that people are working on, this is really not a concern. It's things are held. Usually, even in most, despite there not being an international agreement, most countries are quite good about restricting what can be done and what happens with the things that get produced.
Yeah.
So it's not something that I think people need to be tossing and turning in their sleep about right now. It's most of these are sort of future questions for the future.
Exactly.
We're getting to be able to do more and more stuff every day, so we need to be thinking now about what we want to have, what controls we want to have place in the future as we get better at manipulating genetic material.
I think that's always, those ethical discussions are always a good thing to have in the classroom
Yeah,
as well with
sure.
the younger,
For sure.
middle and high school
Yeah,
students.
absolutely.
Because I think that in general, when I've asked my students before about who polices the
Right.
scientific community, and,
Sure.
and I think they just think, oh, well, scientists are held to a higher standard because
Right.
they're scientists, they're professionals,
Right.
but there
Yeah.
isn't a
That's right.
a police.
Yeah, we certainly
Yeah.
hope so, but
Yeah.
that's right. We're sort of our own police in most
Absolutely.
cases. So
Yeah.
so that's really important. I think I absolutely agree that having those discussions from the beginning all the way
Right,
up is really critical. It's an important topic.
I appreciate you brought it up. That's good.
Just because I want some bamboo in my backyard doesn't mean the
That's
entire
right.
state needs to be covered
That's
in
right.
bamboo.
That's
true,
Because we've lost our maple trees, no
That's
maple
right.
syrup.
Yeah, that's right.
You know? Yes.
Yeah.
And so those are things that I'm glad that, we have the ethics committees, and I'm
Yeah.
glad there are some
Right.
checks in place in different
Yeah,
places.
Right.
And hopefully, eventually, we get to an international.
Yeah. I think
Something like
it'll be
this.
coming before too long. But we're not quite there yet, but I think it's coming. So.
Fair enough.
Interesting. Yeah.
And so, what type of other spectroscopy? You just deal mainly in that one narrow band of our electromagnetic spectrum, then?
Well, let's see. So I work in two narrow bands. Let's say that. So,
Yeah.
so in the visible range, so that's between 400 and 700 nanometers. That's what most people think of when they think of light.
Yeah.
So I work in that bandwidth, and then also in the sort of mid-infrared. That's the region in which most protein bonds vibrate. So if you think of two carbon atoms or a carbon and oxygen atom linked together, one of the groups that we study a lot is the CO double bond in the backbone of proteins. So I mentioned that proteins are these linkages of amino acids. The bond that links them is called an amide bond. Why they're called amino acids. And that's composed of nitrogen and then a carbon and then a double bond to an oxygen.
Okay.
And we monitor the stretching frequencies of that CO double bond, and that's what gives us information about protein structure. So is the protein folded in a helix or a sheet or something else?
So what I'm listening is like selfish. I'm thinking, okay, so what do I need for my grow lights? Because it's about garden season,
Yeah.
so we want to get our plants now. I'm like, okay, so what wavelengths am I?
I'm afraid infrared won't help you very much there.
All right. Well, you see those online. You
That's
see
right. Yeah, that's right.
this is better for grow
That's right.
lights.
Yeah,
This,
yeah.
I see some of them because they have infrared in
That's
it.
right.
And things like, oh.
That's right. Yeah. I think visible and UV are sort of your friends up there. So.
All right. Let's go with the most interesting. This is a question we always love asking. Is what's one of the most interesting projects that you've got to work on in your career thus far?
Oh boy.
I don't know. That's like a.
So far.
It always catches people off guard.
Sure. Yeah.
Very rarely do they're like.
Well, so I should probably say, in some sense, the project that I'm working on now because it's sort of a reboot of a project I worked with almost 10 years ago now as an undergraduate researcher. So the reason I'm doing what I'm doing today is because I got involved in undergraduate research when I was an undergrad at Kansas State University. So I worked for 4 years in a research lab there, again studying photosynthesis and in particular trying to understand what the different chlorophylls were doing in the photosystem II complex. This is the one that produces oxygen.
Okay.
So what I'm doing now is actually a sort of more advanced version of what I was working on as an undergraduate back then.
How neat.
Yeah, so it's sort of, I've been a lot of places in between, and now I'm sort of coming home to photosynthesis again. So.
Very cool.
Yeah.
Very nice. So you tell us which plants will like put the most oxygen in my room?
Well, come back to me in about 6 years.
All right.
Great. That's good.
So last thing, advice for students.
Yeah.
I can't remember how we usually ask that.
Yeah, it's
Because you usually ask it, but
students that are listening, if you know, they think,
Sure.
wow, this sounds really cool. I've talked about photosynthesis in science. I'm interested in that. What advice do you have for them to
Sure.
now, right now? What can something they can do now?
Well, if you're close to a university, see if there are opportunities to get involved in research, or even if you're not close to a university, get involved in a summer program like the Project SEED that the NSF program that we talked about earlier.
Yes.
There's a lot of opportunities out there actually for even high school students to come in and get involved in research, see what a research lab looks like, and get some hands-on experience working with these problems. So that's I think the best recommendation I can give.
That's great. And how easy is it for a high school student if they're thinking, wow, that sounds kind of, I don't know where to even begin, to seek out opportunities?
Right. Well, so at Purdue we have this project SEED. I don't have the web address off the top of my head.
Oh yeah. We can include
put that
that.
in the description
you
notes.
could,
we
can help
notes.
out there. But yeah, through the National Science Foundation, NSF, and the American Chemical Society. Oh, Project SEED is actually ACS funded, I think.
It is.
NSF. Yeah,
right.
my mistake.
Oh, that's okay.
Both through the National Science Foundation and the American Chemical Society, American Physical Society. A lot of these places have programs in place for helping people get involved in research.
Excellent.
And yeah, I definitely check those out. Check with your teachers and any connections you have to academia. Usually people are quite willing to help get people involved if you can make the connections. So, yeah.
Well, that's great. That gives us some great resources to start with for kids that are interested.
Yeah.
So.
It really, up till the last year or so, I didn't know, working here in outreach, I didn't realize that high school students could, the faculty would allow them to come in and actually work on projects.
Yeah, I actually, as an undergraduate, I mentioned doing undergraduate research. As an undergraduate, I also mentored a high school student who was working in the same lab. So we had a whole series of graduate students at high school and undergraduate working together in the research lab. So it's actually more common than you'd think. And don't be scared by getting into research. It's actually not that hard. Nobody comes into research knowing what they're doing. Everybody has to, no matter what classes you've taken or what you've done in the past, coming up to a new research project is always a new process. So whether you're a graduate student or an undergraduate or a professor, there's always a lot of things to learn. So don't worry about not having the experience you need to get involved in research. Learn it as you go.
That's great advice, because I think kids hear that and instantly think, oh gosh, that sounds really tricky, or I'm not sure that's for me.
It's actually really easy to get into it. I mean, it takes a lot of work, and and you got to spend some time with it. But anybody can do it.
I think it might be my favorite quote right there. No one goes into research knowing what they're doing.
Yeah, that's absolutely
even
true.
professors.
Yeah, that's right. Yeah. I've switched fields more times than I can think of right now, so it's always fun. Going into something different. So. Yeah,
well, excellent. Well, we thank you for your time.
Yes, thank you.
We appreciate you coming,
Enjoyed it.
letting us interview, and sharing what you do with the world.
Thank you very much.
Thank you for listening to our podcast.
Be sure to subscribe, rate, and leave a review.
An outstanding
on
review.
iTunes or your preferred podcast player.
Tweet us your science questions.
At Purdue SOS.
Until next time, be super, and remember,
You are someone's hero.
boiler up.
Hammer down.